deswegen Graphene, deswegen mit Graphene. Nicht so viel irritieren.
Heute bekannt.
Good morning. Today is to the subject which is subjectively probably most important, namely life. So, what do we need to live? Many things. Let's energy. What else? How we get energy? We don't have, something to stick into us, right? We don't get energy from electricity. Where we get energy? From food, right? So we need to eat and drink. What else we get from food?
Building blocks, like nutrients.
Nutrients,
like
matter. Let's be physicists, right? So we need energy and matter. Do we consume energy and matter?
Yes, we transform them.
Transform them. Yes. But do we really consume means that you take something in return of yours, right? A child does this, right? A child grows, takes matter, and it builds itself. Let me tell you bad news, I don't do this, right? My body doesn't do this. I'm actually now a little bit shorter than I was 40 years ago. I'm fortunately not, that big. So we don't consume matter, right? What about energy? Do we consume energy? Which means that we take in more energy than we get out. No. Let's return to matter. Every matter which comes into us comes out. Every single atom. Now, matter is conserved, right? So if I take in more matter than I take out, then I will grow. As long as I don't grow, I'm a stationary. Atoms could be different, but the amount of atoms which enter is the same as exit. It's very interesting, incidentally, if you study physiology, how actually it exits. Well, there is an excretion system, but you also will breathe, and we breathe very different things in and out, and also it's very interesting and complicated. Energy is the same, or we get out less energy than we take in.
Depends on the definition of letting out energy.
Letting out, incidentally, how we let out energy? Heat. There's only one way usually. Well, we can do work, but essentially the energy that we steal, it's usually heat, right? So you can measure amount of heat that you generate, which incidentally Robert Meyer did, right? And you can feed a mice a food and then measure amount of heat it generates, and you can also take the same food and do oxidization to it. Well, some kinds of food you can burn and just measure the amount of heat you can generate this way. So we eat chocolate, which 500 calories per 100 grams. You don't find such things, such concentration of energy around normally, right? And we generate body heat. It's the same amount of energy in joules. Because energy is conserved. We don't store it. You are getting more energetic with time, right? You don't have inside any battery which you can put energy in. So what we now come to conclude that we take in energy and matter and we get out energy and matter of the same amount. So why do we need it? I'm interested. You get it in, you get out. It looks like. So what do we really consume when we do this? Why we do this?
Keep our body temperature steady.
Yeah, but okay, you can keep your temperature. I mean, this has its temperature steady.
No, it
Absolutely.
doesn't have the temperature steady if the temperature outside is cold.
But temperature outside is given. There is a sun over there. Instantly, the sun is a source of all energy on this planet, right? From wind to our plants, etc. So to keep energy, you just need to be in equilibrium with an environment. The environment has a finite temperature. It's determined by the distance from the sun of the Earth. The temperature of this planet is just determined how much sun radiation comes in and how much sun radiates out. You can keep your temperature without eating or drinking.
No,
To
we
keep-
can't keep our
specific
temperature
and
37 degrees if that's outside temperature.
What's wrong with
Zero.
37? What I mean, so you can keep your temperature 25. It's actually 28 now outside, not that far from 36.
To keep us-
No, it's not about keeping temperature.
To keep us away from equilibrium.
Huh?
To keep us away from
To
equilibrium.
keep us away from equilibrium. Why we want to be away from equilibrium?
Because we want
Be
to
alive.
to not be static. We want to
Because equilibrium is this. My atoms will be in equilibrium with environment not only after I did, long after I did, my atom would take a lot of time to come into equilibrium with an environment. Okay? So we want to stay out of equilibrium, and for this reason, we eat, namely consume matter and energy. How this helps us to stay out of equilibrium?
we raise
We're
the
solving the problem of life. I want you to really concentrate.
We create order.
Huh?
We create order in our body.
Yes. Order is one of the definition of staying out of equilibrium.
Yes.
Equilibrium corresponds to a maximal
So we're
disorder.
Lowering yes.
What we lower?
We lowering the entropy.
Yes, we lower the entropy because equilibrium is a maximum of entropy. Any one of me and you are out of equilibrium means that we have a lower entropy. Why do we need to eat to get lower entropy?
because the total entropy of the universe rises. So to lower our entropy, we need to raise something else's entropy.
Absolutely. How we do it? Let's take a plant, a much simpler object. Okay? So what plant does? It takes a sunlight. That's its source of entropy, of energy, right? It emits the same amount of energy here as a heat. That's what it does, right? And also it takes some matter from air, but then again, if it's not growing, more or less, it's kind of also emits this atom. So what's the difference between energy which comes from the sun and energy which the plant emits locally? It's the same amount of energy.
It's the chemical energy that it stores.
Chemical, forget about it.
It's localized.
Energy. Like you said, we need to lower entropy.
It's localized.
Localized, sun is localized there, we're localized there, but there is a great idea of this lady is that we need to lower our own entropy. How we do it? Like you say, we need to get entropy out. Okay, which somehow suggests that what plant emits, what our body emits, it's a high entropy. We take entropy out.
We take it very
But energy is the same, right? So energy is the same, but entropy, what we get, must be lower entropy than what we get out must be higher entropy. That's a way to have our own entropy lower, right? We eat entropy, right? So let's see how we do it. We eat a negative entropy, right? So we always produce entropy, which means that we eat. What is minus entropy? Minus change of entropy?
Information.
Information. That's why the subject of this lecture is information theory of life. There is a famous quote of Richard Dawkins who says that what is inside every living being, not a warm breath, not a spark of life, it's information. Okay? So, take a plant. Okay? So there is a sun somewhere. It gives its energy and then it emits this energy. What can we tell about the amount of entropy in the sunlight comparing to the amount of entropy emitted by the plant? Again, we started on the first lecture from this. Well, imagine, well, it does some work pumping water, but just for simplicity, that's our environment, that's the sun. What's the difference between them?
Work.
Huh? No work. I mean, imagine just this plant now just gets some sunlight and emitted some heat. But what's the difference between, whereas you emitted this, what's the difference between this reservoir and that reservoir? Huh? Yeah, but why is the difference entropy? Huh?
It's the temperature difference.
Of course, it's a temperature difference. It's a very, very hot object. Which means that here, when you ask how much entropy this guy consists, it's this heat divided by this temperature. And then when you ask how much entropy this we emit over here, this is delta S1, this is delta S2, it's Q2 divided by T2. Even if Q2 is a little bit less, because you did some work, this is much, much, much smaller than this. Which means that this is much, much larger. Okay? You see the pattern? Sun is a low entropy source of energy. For the simple reason that its temperature much, much higher. It's very simple. Most of the students, anywhere I teach it, I mean, Harvard, NYU, Weizmann, you know, everywhere people are surprised and you must be surprised. It's a really amazing statement. But physically it's trivial because entropy is energy divided by temperature. So sun is a low entropy. Low entropy means kind of ordered from our viewpoint at our temperature. The same with the chocolate. What is 500 calories, kilocalories? I mean, well, it's painful when you try to lower your weight, but it's amazing feat of chemical engineering, right? To put 500 kilocalories into 100 grams, you need a lot of information processed. It's a very structured matter, right? A good steak is also a very structured matter. You know, cow took a long time stay out of equilibrium producing this piece of meat. And what comes out of our excretionary system is a very, very high entropic, low ordered matter. Okay? So we are absorbing information. That's what we do. That's why we eat. That's why we get matter and then we have energy. Okay? Is it clear? Questions? Think about it.
I still don't understand why you said, like, I understand the math, but it's still weird that the sun, you say this, is the sun itself in low entropy because the sun, it's huge and blah, blah, blah.
No, but I don't know what is entropy of the sun. Sun is a huge object, and if you say that entropy of the sun is my uncertainty about it, then it's determined by the total amount of atoms, etc. over there. In the last lecture, we'll talk about the entropy of black holes and how much entropy could be in a given. What I'm saying is about entropy change. If you get some amount of heat of sun, the same amount of heat you radiate here, the entropy you radiate here out of your body is much, much larger than the entropy in the sunlight which you use to trigger your processes. Okay? So we interrupt motion of energy from low entropy, just a second, from low entropy source to a high entropy sink. We put ourselves in between so that more entropy comes out that goes in. That's our way to stay out of equilibrium, means alive. Again, the question?
Yeah, is this somehow manifest in the temperature dependence of blackbody radiation? Like if you look at how
What radiation?
the blackbody radiation, the thermal radiation. Like, is there some way of looking at plots of thermal radiation to see like that this
Well,
is
in
somehow
a
lower
sense,
entropy?
I mean, this is black body radiation at this temperature. This is, you can consider also black body radiation at that temperature. Temperatures are very different, even the spectral curves are very different, but I'm not interested in, at this point in any spectral characteristic of this radiation. Just the fact that if you really take out of sun this amount, you lower the entropy of sun a little bit. And that energy you get into you, that energy you get out, and that means that you really emitted entropy outside. You increase the entropy of the earth, you decrease the entropy of you inside. You eat a chocolate, and then you emitted the same amount of energy as the body heat, you decrease the entropy inside you. Okay? Well, this is now just a phenomenology. Of course, there are many, many very complicated processes happening inside, which stays you ordered. Namely, I mean, I'm not dissolving, green protoplasma in front of your eyes, but that's a basic phenomenology. Okay. More questions? Is it clear? Yeah. Because now you have an instrument, information approach, and I want you to use this instrument for many, many things. I'm just showing you how you can use it. Now, let's see how it works. For example, what I really, let's see. I want to produce protein out of DNA. Okay, you know how it's done. What kind of mechanism our body use to reproduce a structure? For example, protein is a very structured macromolecule. The information about it is encoded in DNA. Okay, so we have one important way of encoding. Oh no, that's terrible. One important way of encoding information. They're all terrible. This. I'll stick to Greek. What it does, it uses the principle of complementarity, right? So the complementarity means that there are these bases, which we discussed because there are 4 bases, and there are triplet code, which encodes amino acids in a protein. So we may ask, what is the typical energy of, it's actually hydrogen bond, it's type of chemical bond, which if you compare it with temperature, it's about 10 times temperature. That's a typical energies in which most of these molecules operate. Sometimes a little bit more, sometimes a little less, but this more or less. Now, it means that there is my kind of ribosome. I don't want to make sketches of which I understand little, but kind of you have something and then something else need to attach to it, and this want to be attached to this because the energy of this attachment will be larger than energy of that attachment, of misattachment, right? So what is the probability of error in such and you have a soup, right? You have a solution where many monomers and bases and all this float, and then they need to start attaching to each other. So you have this this pattern on which they need to sit in the right places. But if it sits in the wrong places, what is the probability of it?
Ratio of exponents.
Huh?
The ratio of the exponents of the energy, like Boltzmann.
Yeah. You say that it must be Gibbs estimate, something like this, right? So it's, as long as you have E over T, that's your Gibbs factor, which tells you more or less what is the probability. So if you now, what is this in terms of numbers? It's E minus 10, which is approximately 10 minus 4, right? Is it large or small? What's your feeling?
No,
Huh?
it's a large error rate
It's large. Why do you think it's large?
because the DNA is very large.
Because, you know, a typical protein is 300 amino acids, which means 1000 bases. So if you make a mistake in one basis, you make you have 10% of your proteins just, you know, with defects. That's lethal. You cannot live in such situation. So, but this is an equilibrium estimate. Would we be in equilibrium, then the probability to find out right matching and the wrong matching will be like this, because this is a Gibbs distribution. This is a distribution of a body in the equilibrium with a thermostat of temperature T. But we are not in equilibrium, and this precisely already on this level shows you how actually being out of equilibrium helps. It provides for error correction. Namely, if you have your DNA and somehow a wrong piece of macromolecule attached in the wrong way, there is a mismatch, it has a way to correct it. How would you do it?
Redundancy.
Huh?
Redundancy. Redundancy.
How would you use redundancy? Well, we know that first of all, you want to do error correction, you need redundancy, which means you lower your capacity of information transfer. You are now doing information transfer from DNA to protein. Because that's what we do, right? All the molecules are there. We just do information transfer, namely the ordered structure of DNA is getting transcribed in the ordered structure of amino acids. So we need redundancy. Good.
You make sure that one mistake wouldn't destroy the protein.
That's what we already did. Incidentally, you remember, we have 4 bases, and they are in a 3-word, every 3 word means, which means that 4 in the power 3 is 64, and we have only 20 amino acids, which means we already have triple redundancy. One mistake in this often does not mean. And yet, this is on a level of a basic code. But there is another level, is just to correct error. Namely, not to use this molecule with the wrong position.
If there is an error, as long as the wrong connection stays, nothing happens with this protein until at some point it breaks and then it can reattach at the correct place.
Fantastic. I mean, what you did now, well, maybe you read somewhere, because sometimes, you know, when I have a brilliant idea, so I probably read about it 5 years ago, and now it just came out of my brain. It's called kinetic proofreading. For some purely technical reasons, these 2 people did not get a Nobel Prize. I don't want to go into this reason. They definitely must be. So the idea is called kinetic proofreading. And I just want to mention it, even though I'm not teaching you biology, it's just, this is a piece of information science, right? So essentially what you do, if you get some kind of, you know, misaligned and something kind of right aligned, you let it stay, not using it immediately. So you have this ribosome, use DNA coming inside it. Actually, Ada Yoran received Nobel Prize for exactly visualizing this process, how a ribosome works. And the molecule goes in and synthesizes over there. But you're not immediately using this, you delay it. You delay it and you use extra processes. That's precisely what you do in non-equilibrium. So you push it in kind of some direction. There are other molecules. I'm not going to chemical details. It's called ATF, adenosine triphosphate acid, and it comes and it does what is called phosphate hydrolysis, which is again not important. It kind of makes it live for a while. Now, if something has a higher probability, namely, this has a higher probability than this, right? It's just because its energy, the energy of attraction is larger, which means that, okay? So your delta E is kind of, but it also means that from this state, it decays back into undetached state, and it also can dissociate. this rate of dissociation is faster. It's called detail balance principle, that if at any state has a higher probability, then the rate of escape of this state or transition, I won't have time, but in the book there is a whole chapter about random processes, random walks, and it has a section which is called detail balance. You've learned it, physicists among you learned it in physics, I'm not teaching you, but the whole idea is clear, and that's precisely this idea, that if you let it stay for longer, then of course you would lose part of these molecules, and you would lose part of these molecules, but you would lose larger part of these molecules. Okay? And that's the idea. This way you do lower your rate of information transfer. And there's redundancy on a different level. Not the redundancy in the level of encoding, but a redundancy on the level of transcription. This is your channel, right? So your information comes from here, or you can say from here to here. But you lower this rate intentionally by letting them dissociate, because this dissociate fast. Okay? And what essentially you use, you can say that of course this ATF is an ordered molecule with a lower entropy. So you can say that every time you multiply this probability of error by something which is a change of free energy, right? And if this could be something like 10% per protein, well, when you multiply by the number of proteins, but then when you multiply it, you would make it squared. So you turn your 0.1 into 0.01. So you can 10% into 1%, because it's factorial. And that's a whole great idea of kinetic proofreading, and it tells you how actually we stay out of equilibrium correcting errors. Okay? So this is on this level. What other system we have of information transfer and control which keeps us out of equilibrium? One is genome. Genome is a fantastic system. You know, a typical bacteria is 1 million bases. Okay? So, there are 4 bases. So you take 4 in the power 10 to 6. That's in a one bacteria a potential number of genetic combination. It's more than the number of atoms in the universe. It's fantastic. I mean, and potentially every combination makes sense, right? Because it corresponds to some kind of a code which in principle may be useless for this organism, but will be useful for other things. Okay, so it's a fantastic system which is a very well, with high fidelity, as they say, right? That provides high fidelity and also potentially infinite number of combinations. What other system in our body keeps us ordered?
Maybe the immune system? Immune system?
I'm talking about information. If immune system uses it's again like this is all is a you know cells, organs, etc. But there is something which directly processes information.
Nervous system?
Huh?
Nerves?
Of course, this organ, right? And most of it, of course, it's not about thinking. It's about keeping our organs, etc. Incidentally, this is clearly digital, right? Discrete units of information, right? Everyone is a big molecule, but it's still discrete, right? You can write it in bits. How our brain, is it digital or it's analog? Namely, discrete or continuous? What's your feeling? Huh?
Analog.
Analog. Which means that it's continuous. It does not count information in bits, but rather, why would you say that? How it does it?
Because the weight of the connections is it's continuous now.
But what is brain activity?
They either shoot or they either fire or not fire.
What neuron does, it either fire or it doesn't, right? It's definitely digital, right? And that's, so for some time people thought that, and when we speak, alphabet, all this, what we discuss, is clearly digital, right? On the other hand, you're right too, because these pulses, you can encode information in why there is a pulse or there is no pulse, right? It's from engineering viewpoint, it's an ultimate case of amplitude modulation. So your amplitude either 1 or 0. But there is also frequency modulation where you can encode your information, and there's a frequency of pulses. And this is continuously changing. Our brain uses both, and continuous, and this in a very, very subtle and complicated patterns. Okay? Good. So now, what I really want to discuss with you, how actually brain processes information. But we start from a very, very simple question, namely a neuron which just attached to your eye or to your ear. For those who were on tutorial last week, I talk about more complicated things, how actually we perceive information, the perception problem, active inference approach. It's very, very speculative, and so I did it on tutorial for brain researcher. But now I'll tell you something which is experimental, really things that one can make. Okay, so I see something and I perceive it. And you can ask simple questions, for example, and do simple experiments. So, and this experiments were really done at 50s, very famous. By now, a person is shown a number, and it tells 5 or 6. Okay. So, what could you measure in such type of experiments? You are natural scientist. Right? So, imagine that you are trying to see if all this information has any relation to how really our brain operates. The simplest brain operation is a recognition of a pattern, of an object. Okay? So you can show different objects, and the person recognizes them. Let's say he recognizes it correctly every time. So what could you measure?
How long does it take?
Time. You can measure how long it takes. As long as it's more or less, you give a person all the time in the world, so it eventually correctly recognized, so you can measure time. Good. So you measure time. So you now want to do science. Namely, you want to compare this time with some theoretical prediction and get a Nobel Prize for supporting or disproving some theory. So what would you compare this time with? First of all, you need different experiments measuring different time apparently for different sets of objects. You can make a person recognize a letter, or a 1-digit number, or 2-digit number, or 3-digit number. Apparently times will be different, right? Or if you take a person and make him recognize a beginning of that symphony or this symphony, more complicated cognitive task. So you measure these numbers times. With what would you compare?
Time it takes the signal to get from the eye to...
You know it, you measure it. You measure the time, which is essentially time it takes from your retina to your neuron to get into whatever is inside and give the answer back, send signal to your mouse, and your mouse pronounce this answer. You measure it. Now, with what would you compare it if you just learned information theory as all you did? The actual experiments that people did. Which means people did, namely they thought of doing such experiment.
A single neuron firing?
Huh?
A single neuron firing? To the average time?
We'll talk about single neuron firing. That would be about capacity of a neural neuron. That would be second part of today's lecture. But okay, 1-digit numbers, 3-digit, 2-digit numbers.
To the amount of bits of the object that represent the object.
Because you learn information theory, right? So it must be something about bits. Okay. So let's say you are a theoretician. I'm a theoretician. You experiment. You measure this time. You come to me and you ask for prediction. Okay, and I don't go into your lab. I'm a theoretician. So, but I'm making a prediction. What kind of prediction you would make? How much longer it would be taken recognizing one of two-digit numbers from one-digit number. There are 9 with 0, 10 one-digit number, and there is 99 two-digit numbers. Okay? So you recognize one object out of 9, which must be faster than recognizing one object out of 99. Make a prediction. Make it now without seeing.
Log.
9?
Factor of 2.
Factor of 2. Somebody else, or factor of 10. There is factor of 10 between, here is 10, 100, and there is factor of 2, means a logarithm. Okay, so is it log or is it n?
Log.
Log, yes, we learned it. So, because what is this log n? N is a number. It is a measure of uncertainty, right? Or how many questions you need to ask to get into one number. And it's proportional to the number of time our brain really operates. It's called Hyman law. Okay? But you cannot assume it. You need to go and measure, right? So you see, this is just early 50s. Namely, Shannon is 48, and people immediately read it and started to ask, okay, now I know the log n, how I can apply it. And this guy went to the lab, put people in front of it was not even a screen, it was just showing them, these numbers, and he measured it. That's how it works. Yeah? You read some paper, you immediately ask, how I can use it? I would check if my time. Then you can do it a little bit more sophisticated. Namely, you start showing some numbers more frequently than others. What do you expect the reaction time to behave?
The average reaction time.
So you have n, you have two-digit numbers, but now you show some of them more frequently.
Faster.
To be fast. Why?
The brains are all adjusts to their probability of the law.
Lower,
We have
lower
more
entropy.
information.
Low entropy. So you do the same measurement, namely, you take minus pi log pi from I1 to n. And by convexity property, as long as your pi is not 1 over n, this is strictly less than this. And they measure it. So you measure, which means that it's fantastic because the moment the person starts, it's still log n. In like, I don't know, after 15 objects. It's not even the whole statistics. The time started going down. And eventually it saturates when you already have a typical sequence of it. Our brain is fantastic in recognizing patterns. And of course you don't even notice it. But the fact that some objects are more frequent means that the entropy is less, means that uncertainty is less. And time goes down. Good. Now imagine on this very subject, but experiment which turns the table. How would you turn the table? Incidentally, always ask yourself when you're doing some research, could I turn the table? It's very, very kind of useful.
He's biased. You can check if he's biased for one of the numbers, because if the entropy is lower when you just give it.
What do you mean it's biased? I mean, you prescribe the statistic yourself. I mean, you experimentalist, you can show some faster.
If the probability of the numbers you're showing is equal, but the answers he's giving is not equal, then you can know he's predicting something about the frequencies.
Yeah, it could be, but our brain, if the probability are equal, our brain very fast learns it. Incidentally, you can also do conditional probabilities. Namely, you can show one numbers more often after some other numbers. Again, lowers entropy. Okay? Again, it lowers entropy. Conditional entropy is always less than unconditional. But you can turn the tables, really turn the tables.
Error rate?
Huh?
Error rate?
Well, errors in the moment we come to errors. But you can do experiment in a different way. How would you do experiment in a different way? Because before you measured the time, okay? You've given all the time in the world, the person answered, and you measure how much time it took. What you can do as experimentalist?
Set the time.
Set the time. Yeah, that's a great idea. That means turn the table. Good. And you did it independently. So I witnessed. So you really make a bum or you make a sound which requires a person to answer within a finite time. As a result, there will be some errors, right? Because somebody did not yet come to this recognition, so you need to guess and make a fast. That's how you train intuition. Okay, so you prescribe time. So what do you measure? What should be linearly proportional to this time? Now you can change time. You can see how the number of errors decreases as you increase your time. Okay? So what would you measure which would you expect to be linearly proportional to the time you set?
Logarithm of error rate, like minus logarithm of error rate or something like that.
So you have i's, or I don't know, which is the true numbers. You have j's, which is what the person say, and they are generally not the same. They are often the same, but generally not the same. So there is whole statistics, pi of ij, and you can measure it, right? What is the probability of observing this pair? You show 3, he tells 8. And that happens, you know. And then what? What would be linearly proportional to your time? Yes. Must be entropies, right? Must be something entropic, because we already have these two entropic. It's a very famous graph. It's called Hyman-Hicks law. Sometimes Hyman, sometimes Hyman-Hicks law. But this is really even more beautiful, right? So you have these statistics. What you can get out of it, which would be linearly proportional to the time. What entropic measure? Because this is about recognition, right? So you recognize, namely you've got this, so there is i's which come and j's which you decide.
Capacity.
Capacity is a very, very right word, but capacity is a, what incidentally, let's recall what is capacity. Capacity is a maximum of a p of i's, in this case input.
And the number of digits that you can.
And what stands here, maximum of what?
Future information.
How you measure mutual information? Well, you take this, you can also out of it compute the p of I, compute the p of j, compute s of ij, compute s of I, compute s of j, and take the difference. This would be your I. That's linearly proportional to the time that you set. Okay? So this is and was really important things that what we invented, I mean we, Boltzmann and Gibbs and Shannon, I mean these formulas is really how brain operates. Because again, as in 20s, people were suggesting very different measures for information, right? The formulas came from 19th century, but using this for trying to quantify something which is really information is about our brain, right? And then people experimentally has shown that these formulas are really related to the way that brain operates. And in particularly, what they've shown that the time is proportional to the amount of information. Okay? Every time you recognize the pattern, you remove uncertainty, and information quantifies the degree of uncertainty. Okay? So let's make a break, and after the break, we'll see how this amount of information divided by time, which is the rate, how it's maximized.
Say 1 digit or 2 digits, but like because then you could say like it's just proportional to the number of digits they see. Like they did it also for like let's say only 1 or 2 or 1, 2 or 3 or 4. Like when you have the same number of symbols. But
Don't no, they were. I guess 1 digital was minimum, namely 9 was minimal, and usually they work with larger because you want to see the difference on a logarithmic scale, which means that you need, and there were like 10 points, which means that it's in logarithmic scale, like 10 decades. But they also did other things. For example, in a restaurant, how much time it takes to find an entry, and if the menu is ordered, it's logarithmic with length. If the menu is unordered, it's linear with length. So it's also recognition, because digits, we have this positional system which we discussed. Yeah, I described you how this great invention of positional writing numbers in a positional system with algebra appeared. And it means that it's a structured encoding, and that's why you recognize it logarithmically, because you do it by subdividing, right? You see 49, so you're already not asking yourself it will be 56, it's 40 something, and then you recognize. So you do subdivision, and that's what makes it logarithmic. But if you are given an unordered set of objects, for example, in the menu in the restaurant, you shuffle, desserts and entries, then it's linear. Time grows linearly with the size of a menu. Okay, so just to finish with it, we will now when we look at the original data, right? So when we say that the time was entropy, we can say that, of course, that was entropy, but it was really mutual information between I and J, and I was shown, and J what was recognized, as it is an entropy of Ij minus conditional entropy of I conditional on J. When you don't make errors, this is 0, right? Which means that you recognize that there is no uncertainty left after you've seen the object. Okay? But generally, when you make short time, uncertainty appears, which means that it's now mutual information. So the final answer to this, what determines the time of recognition, is the mutual information between the signal into the brain and the signal which hits your retina. Okay? So now what I want you to think, and what we consider in the second hour, is what actually happens. How our body or God Almighty designs this system, right? It's eye, it's ear, it's touch. We have different senses. And so what I now want you to imagine that you are God Almighty, and you on day 5, and you designing animals, right? So what you need, and you are designing perception system, right? What means design perception system? It's designing response function. Let me explain what is it. Okay. So you have, oh my God, like everything. Let's stick to green. Is green visible? Okay, green. No, this is bad green. Oh yeah, maybe this one. So you have a signal. Let's call it X. Okay, and you have your Y, which will be a current which goes through your neuron eventually. Like, so what actually comes in, right? And generally, our body and it kind of has a gradation, like weak signal, medium signal, strong signal, right? So you really want to say, and then the interval of your possible signal coming out. So, for example, you recognize sounds from this intensity to this intensity, or you recognize light from this wavelength. This is visible light interval. So there is some interval. Okay. So what you wanna do? You wanna kind of give some kind of a curve here. I mean monotonic, apparently, which would tell you that at this level you are. I mean below this level you are at weak signals, right? From this level to this level, you are in the medium signals, and when you pass through this level, you switch. I mean, your brain gets a signal that it's a strong signal, right? So essentially, what you need to design is for all sensory systems. Now I want you to think how would you design. Apparently, there could be kind of a linear way. Or there could be something which is non-linear, means that convex or concave, right? So another way you can do this, what this will do. This would say that weak signals are most important, right? So already over here you switch into intermediate level. Already over here you switch into strong. Over here, right? You switch into strong level, and all this system you would treat the same, right? Or you can look and take like, my God, I mean, really, maybe the red one. Oh, this is definitely not. And this would be totally different. You say, I don't care about weak signal. I treat all weak signals. Maybe let's. Well, I probably need to put here 1, 2, 3. So this will be 1, 2, 3. Okay. So here, if I do this, I say all the way to here, I treat it as weak or medium, right? So all these signals, I don't distinguish between them. Okay. So now you see, you have three strategies. One more or less proportional. They're all monotonic, namely, the stronger the signal, then at some point you switch into next level. But one is emphasizing weak signals. This would be like this. Another emphasizes strong signal. I mean, emphasis is a derivative, right? The sharper this curve means that you really grade your thing. Or there may be other ways. So let's start from our system. Why we have two ears instantly?
To echolocate?
Huh?
To echolocate?
To know directions from which sound came. Right? Our brain measures the difference. Okay? So what would you choose for your ear?
For the response to intensity or for
Yeah. I mean, what I am asking, linear, concave, or convex? That's a simple question, right?
Concave.
Concave, this one. So you don't care about weak signals, you only care about strong ones.
I say
No,
convex.
it's the weak.
Ever heard word decibel? That's actually how engineers measure sound intensity. What is decibel? Somebody knows?
The log of the intensity.
So why would we somehow characterize the intensity by its log?
Because our ears small.
Because our response function is this one. It's logarithmic. So with very good accuracy, it's logarithmic. It means that our brain really don't much care about strong signal. You don't want to be scared, by some lion roaring over there, but you really want to hear if there is some predator or there is some water, very sure there is some brook over there. So what we do know experimentally is that ear works like this. A touch. Just try to put needle into you. I mean, come on. It's really strong signals here. A touch is more like this. Okay? Incidentally, we also have nose. Why we have two nostrils? Well, we have two eyes. Incident. Two eyes. What for?
3D.
3D. What we talk about at tutorial is that our brain has this fantastic ability, with one eye closed, to distinguish photo from a real 3D image. You know how it does it? You wanna use your phone, you can use it anywhere because there is a video of the lecture anyway. So how our eye can distinguish with one eye a photo from a real 3D picture?
Try to
think it
focus at
jitters.
the same different distances.
You've been a tutorial. Our brain never only sees, he always imagines and compares with sees. Our brain knows for some reasons, which, I mean, kind of small hints, because what is the difference? Of course, you can distinguish photo from it if you just move your head. Okay? So just moving a little bit of your head, you can really distinguish. Okay, so scientists are cruel people, so they fix the head of a person and show him a photo and the real 3D picture in the same frame, and it still distinguishes because it imagines what would have happened if we would move its head. How we do it? Well, we do it. Good. So two eyes we need for depth, right? What we need two nostrils for?
It comes to directionality.
When I was teaching this course a couple of years ago at NYU, there were faculty coming, and one famous mathematician, he is now chairman of Mathematical Congress, Jalal Shatah, from Lebanon, incidentally. He said that the most important thing that he learned in last year was in Green's course, why people have two nostrils. And this is a question of information here. You know Jalal, right? Yeah. Okay, so why we have two nostrils? Look, this is really important, guys. You all have two nostrils.
Becca.
Huh?
Becca.
Becca. Maybe you just
We breathe,
when you have a
one
flu,
nostril at a time.
I mean usually both are blocked.
We breathe one nostril at a time. It changes the dominance of the nostrils.
Absolutely right. Because our nose is for two functions and not one. Which two functions our nose?
Smelling and breathing.
Smelling and breathing, of course. Which means that for breathing you want a fast flow, but for smelling you want a slow flow. Believe it or not, it's very recent discovery, and they still argue about it. I have a friend also at NYU who have a lab which is of our former student actually. He did PhD here in physics and now he's professor doing olfaction, namely smells. So one nostril is usually fast, another slow. That's why it's so difficult when we have a flu and only one blocked is already a big disaster. Okay? Good. So now you know the most important piece of information you got from my course. So now let's talk about vision. So this is sound, this is touch. What would we take for vision? First of all, what is special about our eye is that most of the information about external world we get from it. Right? It's really, of course, it's nice when somebody touches you and all this. There are a lot of nice feelings about it, and also hearing is important. But eye is a king, right? Over 90% of information, 89 depends. So what kind of response function you would design for it? It's just simple thing. You have a neuron, right? This neuron sends electric current. You just measure this current, okay? And you compare it with a signal. Signal, incidentally, again, experimentally found, is a contrast. What determines the current is not an intensity of light. It's a contrast between intensity different pixels. Okay? So you have a given pixels, you have a neuron coming from this. There are many neurons coming from different parts of a retina. You can take one of them and measure it. And this guy by the name Lovely did it. And then you can compare it with a signal. So he measured this. This is the current through the neuron. And this is the signal which coming on. And you can change the signal. Right? You can carefully calibrate. So he measured it. What kind of a curve he measured?
See differently in the dark and when it's very bright light.
More or less,
the same response.
Well, first of all, he did it with a fly. Okay, I don't know, kind of did not use a very wide interval. This is the first application of information theory in biology, very famous work. I'm trying to tell you classical work so that you more or less see how it's done. So your discovery will be done differently, right? You need to know different from work. So this famous Laughlin work, 82, it's kind of relative, but it's really, I mean, web biology, I mean, he measured neuron current inside, okay? And what he found, what kind of a curve? It's monotonic curve.
Convex, asymptotic.
Convex, concave, is it end of your imagination? Can you imagine function which is not convex and concave? And not linear at the same time?
Sigmoid.
Huh? What?
Sigmoid.
Sigmoid. Namely, something like this. Right? It's true. That's what Laughlin measured. So he measured this, something which is really have a very sharp derivative in the middle and not at the ends. Again, look, there are three possibilities. You emphasize weak, you emphasize strong, or you emphasize intermediate. So what he found that our eye emphasizes, namely, really want to distinguish much better between something in the middle. Of course, he had like 10 points here. I can, actually, I mean, look in the book, because in the book I'll just, I cannot resist it, because this is his data, and the continuous line is his theory with no fitting parameters. Just look at it. So there are some points and there is theory. Okay? So what is the theory now? Why it is emphasizing the middle?
Because we want to maximize we want to maximize the entropy, meaning it depends on how the signal is distributed in nature. So
So you wanna not emphasize. First of all, in these cases, it's clearly you favor weak signals, and here you favor strong signals, means that they have of different significance for your body. Which means that there is nothing here was about whatever is the entropy or so. It's just that it's more important for you for purely biological reason, right? So for us, weak sounds are more important biologically. It's nothing about entropy. It doesn't tell you as that weak sounds are really more frequent or less frequent. There is more uncertainty about less uncertainty. Is it just more important? But now when you see this kind of sigmoid, first of all, it essentially tells you that probably it's not about these signals or this or weak or strong being important, because otherwise you would emphasize ends. But what is it about middle that means you're thinking in the right direction? But what is it about middle which somehow makes us thinking, and laughing at his time too, about entropy, namely information transfer. Okay,
Maybe it's the small difference around the average. Because if the signal is
Average,
bigger, we can detect a bit lower bit.
I think that you're thinking in the right direction, but I'm not sure that the way you expressed it is because average is just an average. I mean, you can have a distribution like this, and average will be here. Or you can have a distribution like this, and average will be still here, right?
It's the most abundant.
Yes. So what this great scientist did, he measured abundancy, namely statistics. So somewhere on this the very x-axis, he measured rho of x in this interval, the probability, and it found it like this. Where did it measure it? He himself shown, his flight, the signals, he could show different, with different. And he showed them more or less uniformly, right?
Just went outside and measured.
So he took a camera, video camera, and went where?
Forest. I'm sorry.
To the, not actually forest, there was some kind of a swamp, a bog, where this fly actually lives. Okay, good, and then what? You point a camera so you see some picture. Design the experiments. I mean, I'm trying to make you to follow, to do yourself designing this experiment. Huh?
Just take random directions.
So how you do this or this?
Whatever the fly does.
Okay, so Bert suggests we're going like this. You've seen a fly? I mean, it's mostly flies horizontally. It's easy to fly horizontally. Sometimes it flies vertically. So he mostly moved it horizontally, and this video camera recorded the picture. Then he, from this picture, took a mean contrast per camera and got his probability distribution. Okay? Now, this is one curve, rho of x. This is another curve, let's call it y is g of x. So this is my g of x. Tell me what is the relation between these two curves.
G is the integral form.
So the statement is that rho is g prime of x, or g is an integral of rho of x prime dx prime. Why?
Because you want each probability slice to have a corresponding sensitivity slice, like for 10% you have.
It means that, let me really do it in a much more extended way so that you appreciate how much it's similar to what Samuel Morse did. Really, this fly does similar things. Why it's similar to Morse code? What is the idea of a Morse code? Encode rare object by long things. It's not more frequent object by something really short. So what essentially it tells? It has, you have this distribution, okay? And now what you want to do, you take again in this very interval the curve which is like this, okay? Which means that here you have equidistance interval in which you, this is actually your bits, right? That's how you quantify the signal. So now when you look at this, this would be this part of your curve. When you look at Z, that would be Z part. That's like your idea is that the areas are the same. That's what essentially means this statement is that areas are the same. Area is a cumulative probability. So now, I mean, this is encoding of this, and this is encoding of this, right? And essentially it tells you that, and then you would get this one, which is on the peak, and which will be the most narrow, but it would tell you that you, in your transmission, would be transmitting signals from different regions of X with the same probability. Because this probability, I mean, this is your one kind of encoding A, B, C, D. So A and C and D will happen with the same probability because they correspond to the same areas here. And the same areas means that you do it, right? So, well, technically, of course, what you do, you need to maximize the mutual information between X and Y, right? This is what is you do, which is, you can say that this is your S of X, which you can compute out of this row of X. Well, let's write it in the form. It's S of Y minus S of X under a given Y. Okay? So this is one way to write mutual information. Now, at this point, Laughlin assumed, and then people generalized it for more general cases, that as long as X is given, Y is given too. Namely, that there is one-to-one correspondence, which we essentially assume, because when you make this curve, it's a curve, right? It's not a diffuse cloud of points. That means that this is 0. And so this is just S of y. So S of x is given. So you're looking for transform from x to y, which would give a maximal entropy. What distribution on a finite interval gives maximum entropy? Uniform distribution. Indeed, if you say that rho of x dx is rho of y dy is rho of y dy dx dx, then you find out that the rho of y is rho of x times divided by g prime. So if you want rho of y to be constant, you want g prime to be proportional to rho of x. Okay? That's the whole idea. So now, if you transform here into rho of y, you will get this. This is the same principle that you want to do. Again, Morse did it for the same reason, to increase the information transfer in the Morse code, right? So because the more frequent letters are encoded shorter, you increase the rate. What is done here is that you really use all the intervals of your signal more or less with the same probability, which means that whatever signal from whatever interval came, you are maximally uses it. And technically, it's expressed in the statement that you make your probability constant. Okay? Is that clear? And again, as I've shown it here, there are no fitting parts. There are error bars, of course, because you measure things with a finite errors. But what is most important is that there are two things. One measured in the lab, and it's a current as a response for density contrast. And another thing measured outside of the lab in the natural habitat of this fly, and it corresponds to the statistical distribution. And in this sense, it's a direct demonstration that the biological system designed for, and our eye is something which is designed for processing maximum information. That's why it's so crucial for an eye, really, this response function, because it's really neat. We don't know what is important, low contrast or higher contrast. There are too much information of very different nature that our eye processes. It's for brain to decide which is important and unimportant. In a tutorial, I talked a little bit how it does it, but what really the first step is to make a transformation to a constant probability. Okay? Questions? Everything is clear? Or everything is not clear?
Is there adaptability? Like, can we change this curve depending on which environment we are in? Or not?
Apparently, it must be, as long as individual, it actually takes some time, but if climate change or if a given species relocate from region to region, then apparently the response function will be changing according to me. I'm not sure, I'm not in evolutionary biology, so I don't know how much time it takes, but apparently it does. What is evolution? Evolution, no, Darwinian evolution. You know, what is the Darwinian evolution?
Survival of the fittest.
Survival of the fittest. you agree with this definition of Darwinian evolution as survival of the fittest?
I agree.
You agree. Somebody disagree with this definition.
Two different effects. One specific species that's changing because he moved to another environment, because climate change or something, and there's like something happening
What is
from
it?
generation to
Evolution.
generation.
Evolution means survival of a fittest. Namely, that you say that evolution is that this survives and this not. Really have bad news for you. Nobody survives. So what is evolution?
Transmission of information.
What survives?
What genes and
Information is ever improving encoding of information about environment in the genes of its population. No one survives except genes. And gene survives after the whole species already go extinct, not only individuals. Okay? So it's this, of course, Darwin did not do genetic, but also even Darwin had it in a much more. What is really important, not for a given, fittest animal to survive. What is important, it could even live shorter, for him to live descendant, for genes to survive. Okay, as it's a book selfish gene. But your answer is right in a sense that we all think in these terms. But survival of the fittest means survival of the genetic information in the fittest person. Okay. So now kind of takes all this back to square one. Okay, so let me summarize what is today's lecture, and we'll finish today a little bit earlier. Of course, next will be money, and I don't want to, to mix money and life in one lecture. So we started from understanding that essence of life is information. So life is a state of being very far from equilibrium. Being very far from equilibrium means having much lower entropy than your environment, means processing a lot of information, and this processing a lot of information requires a structure, which is structure encoded in the genome and structure imposed by our brain. And it not only requires it, it also uses it for supporting it. Okay? And we also discussed that really our brain operates and our genome operates, genome is not surprisingly, but really brain, according to the same formulas for entropy. And also we discussed the first step of processing information, and this is more or less something which we experimentally are more or less confident. And the things which I talk about, active inference, which is again in the book, it's much more stable. Okay? Quiz 2 until today, but next time please, because there are many people who really did not look. When you look at quiz, there is a date over there, right? So I don't know why system sent wrong reminders, but generally, so from now on there will be 2 more quizzes, and they all will end on Sunday night, okay? Between Sunday and Monday. Okay? So today you still have time to finish second quiz who did not do. Okay? Good.
העיקרון הזה, הוא ממשיך גם בשלב הבא שלנו?
לא. זה מה שאני סיפרתי בטיוטוריאל, שבשלב הבא זה משחק אחר לגמרי. זה comparison of prediction of a brain with what came to. This is just the first, this is a mechanical level. This is done physiologically. Right? Essentially, and what happens in the next, there are like cascade of processing stations. In the smell, it's even more. There are like many come here, and then few comes out, and then many comes. It's a comparison of a signal sent for a brain from a signal sent by a sensory. Our brain is a prediction correction machine. So this what is done there is totally different. Okay? This is the kind of first generation stage. Because I mean you don't do anything to a constant, you already got it. This is the maximal entropy. Now after that you start processing it.
Start lowering the entropy.
Namely, you start playing this game. Namely, you start asking, is it important or not? Which means for this you need to compare it with something. Right? So you have this, what you get? You essentially get your set of pixels, and on the set of pixels you have signals. So you know, so this pixels tell you that it's white, and this tell it's black, that's tell high contrast. It's hugely redundant, hugely. Like it's from judging from how many cells in our retina, it's 10 to 5 bits per second. We can process like 50 bits per second, more or less. Okay, so we need to make a huge compression. For this, I will talk on the lecture. It's called information bottleneck and all this. And actually, self-driving car works this way, right? So the camera get a lot of optical information, and you need to really decide right, left, stop. Okay, but it's not this. There's something else. Okay. Questions?
Small one, technical one.
Yeah.
And the מועד ב' מועד ב' של הבחינה. The second date for
Yeah.
the is it the one that's written in the model? Because I'm in summer school for on the
Everything about exam is so much not final that at the end we all sit together and discuss what is convenient for us. Okay. So it won't be that it's something which is inconvenient for somebody. Okay, that will be all negotiable. Okay.
I have a small question about last lesson, not that story, but the last lesson. When we talked about like when we have these conditions, and you gave again the example of the candy box, like under the conditions that we have. So my question is, in the example of the candy box, we solved it and we got the geometric distribution. But my question is like, if we have a finite number of candy boxes, I expect like, let's say if we have 9 boxes, and we get the average is 5, I expect that we should get like for maximum entropy, which we should get just the uniform distribution. But like I don't see how we can get the like.
It's a good question, incidentally, and I don't know the answer, but you can compute it, right? Because when you get an infinite, then you get a geometric expression.
And this is just like another condition with the function like this.
Well, but on the other hand, I mean, it's clear that if you set mean value, that your probability will be exponent minus g.
Yeah.
And here will be some factor, minus lambda g. How this factor lambda would depend on this being 5 or 6, let's call it r. Okay?
Yeah.
So lambda depends on r. There will be different dependence, but dependence will be still exponential.
Okay.
Okay?
Yeah. Okay, no problem.
As I say, whatever you measure is in the exponent.
Okay. Thank you. This is yours?
No.